Welcome to Module 2 of Introductory Biochemistry. In this module, we will explore enzymes, the biological catalyst essential to life, and vitamins, the organic molecules crucial to metabolic regulation and well-being. We'll also address how imbalances in these compounds can lead to diseases or health complications. Let's begin with enzymes. Lecture 1: Understanding enzymes, nature, structure, and function. Enzymes are specialized proteins that accelerate chemical reactions in living organisms. Without enzymes, life would not be possible, as most biochemical reactions would proceed too slowly to sustain life. Enzymes are highly specific, each one typically catalyzes a single chemical reaction or a group of closely related reactions. They work by lowering the activation energy required for a reaction to occur. Enzymes act on substrates, the molecules they convert into products. This transformation occurs at the active site of the enzyme, which fits the substrate like a lock and key. Some common examples of enzymes include: amylase, breaks down starch into sugars. Lipase, digests fats. Protease, breaks down proteins. Lecture 2: Zymogens, enzyme specificity, and the active site. Some enzymes are synthesized as inactive precursors called zymogens or proenzymes. These must be activated by specific biochemical changes before they can function. For example, pepsinogen is activated to pepsin in the stomach to begin protein digestion. Enzyme specificity refers to the enzyme's ability to select a specific substrate and catalyze its transformation. Specificity is determined by the structure of the enzyme's active site, which complements the substrate's shape and charge. There are four main types of enzyme specificity. Absolute specificity acts on only one substrate. Group specificity acts on molecules with similar functional groups. Linkage specificity acts on specific types of chemical bonds. Stereochemical specificity acts on specific enantiomers or isomers. Lecture 3: Enzyme inhibition and activation. Enzyme activity can be regulated through inhibition or activation. Inhibitors are substances that reduce enzyme activity. They are categorized into: competitive inhibitors, bind to the active site, competing with the substrate. Non-competitive inhibitors, bind to another part of the enzyme, changing its shape. Uncompetitive inhibitors, bind only to the enzyme substrate complex. Enzymes may also be activated by cofactors, which are non-protein substances that enhance enzyme function. Cofactors include metal ions such as magnesium or zinc. Organic molecules called coenzymes, which are often derived from vitamins. Enzyme regulation allows cells to conserve energy and respond to changes in the environment or metabolism. Lecture 4: Factors affecting enzyme activity. Enzyme-catalyzed reactions are influenced by several factors. Temperature: each enzyme has an optimum temperature at which it functions best. Too high or too low a temperature can denature the enzyme. pH: enzymes have an optimal pH range. For example, pepsin works best in acidic conditions, while trypsin functions in a basic environment. Substrate concentration: increasing substrate concentration increases reaction rate up to a point. Enzyme concentration: more enzyme typically results in more product, assuming substrates are available. Presence of inhibitors or activators: these can slow down or speed up enzyme activity. Lecture 5: Allosteric enzymes and isoenzymes. Some enzymes have allosteric sites, regulatory sites other than the active site. Binding of a molecule to an allosteric site can increase or decrease enzyme activity. These enzymes are essential in feedback regulation of metabolic pathways. Isoenzymes, or isozymes, are different forms of an enzyme that catalyze the same reaction but differ in structure and may be present in different tissues. For example, lactate dehydrogenase has several isoenzymes specific to the liver, heart, or muscles. Understanding isozymes helps in diagnosing tissue-specific damage. For instance, elevated cardiac isoenzymes may indicate a heart attack. Lecture 6: Introduction to vitamins. Vitamins are organic compounds required in small amounts to support normal metabolism and physiological function. They are not synthesized in sufficient amounts by the body and must be obtained from the diet. There are two major groups: fat-soluble vitamins A, D, E, and K. Water-soluble vitamins B complex and C. Each vitamin plays a specific role, and deficiency or excess can lead to health issues. Lecture 7: Vitamin Deficiencies and Related Conditions. Let's examine some important vitamin-related health problems. Vitamin A deficiency can lead to night blindness and dry skin. Vitamin D deficiency causes rickets in children and osteomalacia in adults. Vitamin E deficiency may result in nerve and muscle damage. Vitamin K deficiency impairs blood clotting. Among water-soluble vitamins, vitamin B1 thiamine deficiency leads to beriberi. Vitamin B12 deficiency can cause pernicious anemia. Vitamin C deficiency results in scurvy, marked by bleeding gums and poor wound healing. Understanding these conditions helps in diagnosis and dietary management. Lecture 8: Vitaminosis, Hypovitaminosis, and Antivitamins. Three terms are key in understanding vitamin balance. Vitaminosis, general term for disorders caused by abnormal vitamin levels, either too much or too little. Hypovitaminosis refers specifically to vitamin deficiency, often due to poor diet, malabsorption, or chronic disease. Antivitamins, substances that interfere with the absorption or action of vitamins. For example, avidin in raw egg whites binds biotin, preventing its absorption. In some cases, medications or environmental factors can act as antivitamins. Lecture 9: Vitamins and their coenzyme functions. Many vitamins serve as precursors to coenzymes, which are vital for enzymatic reactions. Here are a few examples. Vitamin B1, thiamine, becomes thiamine pyrophosphate, used in carbohydrate metabolism. Vitamin B2, riboflavin, forms part of FAD and FMN, important in oxidation reduction reactions. Vitamin B3, niacin, contributes to NAD and NADP, essential in energy metabolism. Vitamin B6, pyridoxine, is needed for amino acid metabolism. Folic acid and vitamin B12 are crucial for DNA synthesis and red blood cell production. Without these coenzymes, many biochemical pathways would be disrupted. Lecture 10: Biomedical Importance of Vitamins. Vitamins play essential roles in metabolism, acting as coenzymes in reactions involving energy production. Immunity, vitamins A, C, and E support immune system function. Tissue repair and growth, vitamin C aids collagen synthesis, while vitamin D supports bone health. Neurological function, B-complex vitamins are vital for nerve function. Reproduction and development, vitamins A and D are involved in fetal growth and development. In clinical practice, vitamin levels are routinely monitored in patients with malnutrition, chronic diseases, and certain medication regimens. Module 2 Summary. In this module, we explored the structure and function of enzymes, zymogens, enzyme activation and inhibition, factors affecting enzyme activity, the importance of allosteric enzymes and isoenzymes, classification and functions of vitamins, deficiency diseases, hypovitaminosis, and the role of antivitamins. Vitamins as coenzymes and their biomedical relevance. Conclusion of Introductory Biochemistry. Congratulations on completing the course Introductory Biochemistry. You have now developed a foundational understanding of the molecular basis of life, the structure and role of biomolecules, the importance of enzymes in catalysis, the critical roles of vitamins in health and disease. This course sets the stage for deeper studies in medical biochemistry, molecular biology, nutrition, and related disciplines. Your understanding of these core concepts will be instrumental in your academic and professional development in the life sciences.